So tell me, how to actually find out that a plant that you are seeing, which is tall, is actually a homozygous dominant, capital T, or it's actually a heterozygous plant, capital T, small t. How to find it? Mendel was not able to see the genes, and to be honest, we cannot see it even today by our naked eyes. So Mendel found a way to actually find out if the plants, tall plants, visibly dominant plants, are either actually homozygous dominant or it's an heterozygous plant. And that method is called test cross. Let's understand it nicely. So what is a test cross? A test cross is the cross between an individual with a dominant phenotype but unknown genotype, and that cross is done with the homozygous recessive individual. And why do we do it? We do it to determine that the dominant phenotypic individual is actually homozygous dominant or it's a heterozygous plant. And why do we need that cross? Suppose there is a tall plant. That tall plant can have two types of genotype. We already know that, which could be homozygous dominant, capital T, or could be heterozygous, which is capital T, small t. They both look tall, so the phenotype alone, we cannot determine the genotype of it. So we need to perform a test cross. So let's start with the first one, which is the monohybrid test cross. So in the step one, we need parents for the cross. We do not know the genotype of the plant, but we can see it's physically dominant, phenotypically dominant. And recessive plant, I do not need to tell you that it has to be homozygous recessive, which is small t, small t. So the cross could be of two scenarios. So case one, that the plant that we have took is dominant homozygous. How you do it? Simply, you make Punnett square or by Folk method. The capital T will only produce one type of gamete because of homozygosity. Similarly, the small t again produce one type of gamete again due to homozygosity. So the offsprings will be capital T small t, capital T small t, capital T small t, capital T small t. Now you tell me what is capital T small t. A, it is heterozygous plant. B, it is phenotypically tall plant. All the plants will be tall. So the phenotype ratio becomes 100% tall. Now case number two. So the plant that we have taken, supposedly it is a heterozygous plant. It has capital T and small t in them. So you separate the gametes. The heterozygous will produce two kinds of gametes, capital T and small t, and recessive will make one type of gamete, which is only small t. And when you cross them, you will get two different types of phenotype. That will be two tall plants, two dwarf plants. So the ratio becomes 1:1. And that's how you know that the parent that you have taken, the unknown plant that you have taken, is a heterozygous dominant. So if the plant that we have taken is dominant phenotypically as well as genotypically, so if the so the plant that we have taken is of dominant homozygous, so all the offsprings will be tall. Like 100% of the plant will be tall. Dihybrid cross is also performed in a very similar manner. And here the ratio becomes phenotypic as well as genotypic ratio becomes 1:1:1:1. Now, very important concept to NCERT point of view is that A dihybrid cross proves the law of independent assortment. Because if genes does not assort independently or if they are dependent on each other, the ratio will not be 1 is to 1 is to 1 is to 1. So whenever you see that ratio deviates anywhere from 1 is to 1 is to 1 is to 1, it means that it is linked. It is the case of linkage. This is very important question and that goes for monohybrid crosses too. Now, many students do not understand this, that what is test cross, what is back cross. So back cross is F1 generation crossed with either parent. The parent could be either homozygous dominant or homozygous recessive. It doesn't matter. When the F1 generation is crossed with its parent generation, either one of them, it is known as back cross. Okay? And that back cross may or may not be test cross. If it is done with a dominant parent, it is not a test cross, but when it is done with a recessive parent, it becomes a test cross. Similarly, test cross is F1 crossed with homozygous recessive. Important exam point. In monohybrid test cross, if the heterozygous plants are crossed with recessive, the phenotypic ratio becomes 1 is to 1, whereas the genotypic ratio, as you can see right here, also becomes 1 is to 1. Very important, every test cross is a back cross, but not every back cross is a test cross. Now, key formula to remember, please remember that number of gametes in a heterozygous individual is 2 on power n, where n is the number of heterozygous gene pair. For example, you can see T, capital T, small t, only one heterozygous pair. So 2 power 1 is equals to 2 gametes. Here, capital Y, small y, and capital R, small r is 2 heterozygous pair. So the number of gametes become 2 into 2 is equals to 4. Now let's do a recap that what we have studied so far. So we studied that a test cross is a cross between a plant with unknown genotype but dominant phenotype. It is crossed always with a homozygous recessive plant. To actually know what the phenotype the genotype of the plant is. If all the plants come as tall, it means that the dominant phenotypic plant was homozygous dominant. And if the ratio of dominant and recessive becomes 1 is to 1, it means that the dominant phenotypical plant was heterozygous plant. Thank you so much. If you like this video, please like, share, comment. If you have any question, you can ask in the comment section below. And thank you so much.